Hi, my name is Charles Kakilla, and I'm a biostatistician at Tanfia Clinical Trials. And as part of adding to the clinical success guide piece, I'll discuss on the critical roles of statistical analysis in clinical trials. So let's explore what is indispensable and how it shapes the entire process of clinical research. I'll touch on the importance of statistical analysis in clinical trials, types of statistical analysis used in clinical trials, design phase, stats consideration, and finally, handling and analysis of data. So first and foremost, statistical analysis provides an objective method to evaluate the efficacy and safety of a treatment. It is essential for meeting regulatory requirements and obtaining approval from health authorities. So without rigorous statistical analysis, our trial results would lack credibility and reliability. We do use various types of statistical analysis. One way is to use descriptive statistics, which help us summarize the basic features of our data, providing clear summaries and visualizations. Likewise, inferential statistics allow us to draw conclusions and make inferences about the broader population from our sample data. We may complete the multivariate analysis, which is used to examine the influence of multiple variables simultaneously, giving us a comprehensive understanding of factors at play. During the design phase, several statistical considerations are paramount. Calculating the appropriate sample size, for instance, ensures that our trial is adequately powered to detect significant effects. Also, randomization methods, which are also crucial in reducing bias by ensuring random assignment of subjects to treatment groups, is crucial in adding to the design phase. So in addition, blinding techniques as well, which blind the subjects and investigators to treatment assignment, further minimize the bias and enhance the validity of our results. However, it's good to note that this is only available for blinded trials, as others tend to be open label. Now, handling and analyzing the data involves several key steps. First of all, data cleaning and preparation are essential to ensure that the quality of our data is intact before the analysis. We often compare the analysis of intention to treat population with per protocol population in order to handle data from subjects who deviate from the protocol. Additionally, we explore various strategies for dealing with missing data, and these are crucial for maintaining the integrity of our analysis. For example, methods such as imputation, whether it's regression, mean, median, or mode imputation, can be applicable here. And the other method is complete case analysis. This is whereby only cases with no missing values for any variables of interest are analyzed. And the other one is available case analysis, whereby we do use all the available data points for each analysis rather than deleting the entire case with missing values. We've got another method, which is last observation carried forward. This essentially replaces all the missing values with the last observed values. And a couple of other different methods can be applicable to dealing with missing data. But subsequently, selecting the appropriate method depends on the nature of the data, the patterns of missingness, and the specific goals of the analysis. So each method has its trade-offs. In practice, a combination of the method could be used to handle missing data effectively. Moreover, clearly defining the primary and secondary endpoints is critical for the success of our trial. And before we delve into the analysis bits, hypothesis testing should be carefully reviewed as well. This step essentially involves formulating null and alternative hypotheses and selecting the appropriate statistical tests to evaluate them. So understanding not only the p-values, but also the confidence intervals, the effect sizes would help us interpret the results in a clinically relevant context. What is more, when conducting multiple comparison, we must be vigilant about type 1 and type 2 errors, or in other words, false positive and false negatives. Hence, different techniques like, for example, the Bonferroni correction help us adjust for multiple comparison, hence reducing the risk of errors and ensuring reliability of our conclusions. Furthermore, based on our trial designs and outcomes, we often do conduct sensitivity analysis. And we do keep in mind that analyzing subgroup data requires careful consideration. Pre-specified analysis and a defined a priori are crucial to avoid data dredging. Understanding the interaction effects allows us to see how different variables interact and influence the outcome, hence providing deeper understanding and insight into our data. So in clinical trial, one point to add, it is sometimes necessary to conduct an interim analysis or to set off some stopping rules. The interim analysis essentially involves conducting statistical analysis at pre-specified points to assess progress and safety, whereas stopping rules are criteria set for early termination of the trial, whether for efficacy, futility, or safety concerns and reasons. And these practices ensure that our trials are conducted ethically and efficiently. So in conclusion, statistical analysis is the backbone of the clinical trial. It ensures that our research is robust, credible, and variable to medical science. Thank you.